Good afternoon, everybody. I would like to express my thanks for the opportunity to present some results of our studies at this conference. For nearly 40 years, I am a researcher at Embrapa, a governmental Brazilian agricultural research corporation in Brazil. The area of my activities is science and technology of seeds, mainly coffee seeds, that have a complex physiology and doesn't have tolerance to drying and storage. In this context, nanotechnology can be an alternative for preserving coffee seeds, even though our studies are on the beginning. I chose to present some preliminary results in this forum. This study is about the effects of iron oxide and hydroxyapatite nanoparticles on the coffee seed germination, and is part of a project on advanced techniques in cryopreservation of coffee species. Coffee is the main agricultural commodity of Brazil, with strong contribution to the Brazilian trade balance. Brazil is the largest world producer and exporter of coffee. In this last crop year, Brazil produced 68 million bags and exported 45 million bags. This expressive export explains in some way the third place of Brazil in the world coffee consumption. The larger consumer is the European Union, and alone the United States and Japan are the largest coffee consumers in the world. There are two coffee species of worldwide importance, and in Brazil, coffee arabica represents more than 70% of the total production. Minas Gerais, the state where I live, contributes approximately to 50% of the production. It is one of the most expressive cultures in this country and abroad. In spite of advantages in studies on vegetative propagation, fields of Coffea arabica species are still formed by seedlings produced from sowing of seeds, which have low longevity and they lose the potential for germinating very quickly. Given the importance of coffee to Brazil, preserving species is fundamental, whether for farming crops or for plant breeding programs. However, the coffee seed doesn't tolerate storage in conventional seed banks at minus 18 to minus 20 Celsius degrees, as the orthodox seeds are. They are still conserved in germplasm banks in field. That is a difficult and costly operation, putting the genetic diversity at constant risk. Thus, biotechnological alternatives such as cryopreservation are necessary and have been extensively investigated. Cryopreservation is a technique of biological material stored in liquid nitrogen, and in its gas phase minus 156 degrees, or in its liquid phase in 196 degrees. And it is a promising alternative for safe long-term conservation of coffee species. These steps of cryopreservation are drying, preparation of seeds, cooling or freezing, immersion in nitrogen, and thawing of the seeds. Seeds must be dried up to a precise moisture content in order to withstand that extremely low temperatures. After drying, they are immersed in nitrogen where they can remain for an indeterminate time. After removal from the nitrogen, the seeds must be warmed. All steps are important for the success of the technique, but the cooling and the warming of seeds are crucial because they these processes may favor the formation of ice crystals that damage the cells and can make the seeds unviable for the germination. For Coffea arabica seeds, there is already an optimized and validated protocol, which guarantees conservation indefinitely. But for Coffea canephora, Pierre, the second most cultivated species, the cryopreservation is not yet possible. For these and other species, we are now investigating cryopreservation associated with nanotechnology. The big challenge is freezing and thawing without forming ice crystals inside the cells that causes disruption on the membrane systems. This happens because water expands during these steps, conforming ice crystals, especially in the range of minus 15 to minus 60 degrees. In recent years, the use of cryoprotective substances is being used for protecting the cells and can induce a glass state in the intracellular medium, thus reducing the formation of crystals within the cells. More recently, the use of nanoparticles associated with cryopreservation has revolutionized and the cryobiology universe, focusing on the damage due to rewarming. In this technique, animal tissues for transplants are protected with magnetic nanoparticles before immersion in nitrogen. The rewarming performed in a magnetic field makes the tissues warmed in an equally and rapidly manner, protecting them against the damages. This technique was called nanowarming. This maybe can this maybe can be an alternative to protect the coffee seeds during cryopreservation. Given these new tools, the objective was to investigate the effects of nanoparticles during the germination processes of coffee seeds to later be used in association with the cryopreservation technique. This slide shows this experimental scheme, and it was investigated the effects of iron oxide and hydroxyapatite nanoparticle suspensions during the seed soaking and germination processes. Germination of the coffee seeds is a three-phase pattern consistent of absorption of water, phase 1, preparation of the seeds for germination, phase 2, and radical emergence, phase 3. The experiment was performed during the second phase. The seeds were immersed for 24 hours in different concentrations of nanoparticle suspensions and in water as a control treatment in 25 degrees. After they were soaked in germination paper, after that, they were soaked in germination paper during the imbibition and germination process. Samples of seeds were taken for the construction of the imbibition curve and to assess the physiological quality, the effects of nanoparticles. This is the summary of the treatments and the evaluations that were carried out. This picture shows coffee seeds at the two evaluated stages, the root protrusion and the germination phase that shows the hypopotyle emergence. The other picture illustrates the Tyndall effect, the optical path of the nanoparticles in the suspensions. The following tests were performed to evaluate the seeds. Moisture content, of course, in order to construct the Imbibition curve, embryos viability in the tetrazolium test, percentage of root protrusion, of seed germination, of strong normal seedlings, of seedlings with expanded cotyledon leaves, and shoot and roots dry matter per seedling. These pictures illustrates some physiological tests we always perform for evaluate the coffee seed quality. Now, I will present the preliminary significant results. Other analyses were scheduled, but weren't still carried out due to the COVID-19 pandemic. We have the laboratories closed until now. Although a higher seed moisture content was observed at the end of the soaking period, in all concentrations of the both nanoparticle suspensions, there was no statistically significant effect on the seed imbibition curve. There is, however, a tendency of higher seed moisture in the presence of nanoparticles at the end of the both curves that we can see in the curve of hydroxyapatite and iron oxide nanoparticles. In the tetrazolium test, the living areas of the seeds are colored in red due to the reaction between the respiration enzymes and the tetrazolium salt, reducing it and forming a red color compound. The figure shows embryos entirely colored in red and therefore viable and they can germinate. Means with the same uppercase letters in the row do not differ, and the means followed by the asterisk do not differ from the control. In the picture, coffee embryos entirely colored, as I said. According to the ANOVA, there were no effects, positive or negative, of the nanoparticles in the tetrazolium results. The graph also shows a high value of viables in both dispersals and in all concentrations studied. The percentage for radical protrusion can see that these variables indicates the capacity of the seeds for rapid emission of radical. The percentage of root protrusion in iron oxide was lower than in hydroxyapatite at the highest concentration and also lower at water imbibition. Germination at 15 days. Very similar results were observed regardless of immersion in water or in nanoparticles. The seed germination of both nanoparticles studied also showed only differences in the iron oxide dispersion and in the concentration of the 2.5 milligrams per liter. Strong normal seedlings test. In this test, seedlings with hypocotyl length greater than or equal to 4 centimeters are computed, indicating better physiological quality. In this variable, lower percentages was also observed in the presence of iron oxide and in the concentration of 2.5 milligrams per liter compared to the hydroxyapatite or water. For root or shoot dry matter, a bigger variable of the seeds, the values are statistically equal between the two nanoparticles, but higher values were observed for hydroxyapatite at 2.5 milligrams and for iron oxide at 1.6 milligram per liter. The graph does not show a consistent trend as a function of different concentrations. According to the preliminary results I presented here, we know now the nanoparticles don't have important negative or phytotoxic effects, especially at lower concentrations. Other proposed analyses weren't performed yet, but will be done soon. After that, the experiments on the nanowarming technique will also be tested for coffee seeds, especially with nanomagnetic iron nanoparticles. It is expected that at the end of these experiments, it will be possible to recommend a secure protocol for cryopreservation associated with nanotechnology of coffee species of interest. Besides, it is expected the knowledge obtained from these studies may be broadened regarding the sensitivity of coffee seeds to dehydration and to cryopreservation, making information available for studies with other seeds of economic importance for which reliable long-term storage is not yet possible. This is what I brought to present, and I just want to say this study is part of a doctoral thesis at the Federal University of Lavras, Minas Gerais, in Brazil, and the research team is composed of graduate and undergraduate students, professors, researchers, and other collaborators. Marina Chagas is an agronomist, doctorate student and responsible for the studies. I am an agricultural engineer, and this is advisor and the project coordinator. Ana Luisa is agronomist, is a co-advisor and postdoctoral trainee at the university. Other two researchers from Embrapa are chemical engineer and are collaborators in the project. And Luciano Coutinho, an agronomist, another agronomist and collaborator from another university in Brazil. So thank you very much and for this opportunity. And I leave here my email address. Please, all of you, feel free to send me questions and comments. Thank you.